So now we know how to generate acetyl coenzyme A. That acetyl coenzyme A, the product of the pyruvate dehydrogenase complex in the matrix of the mitochondrion, can now enter the citric acid cycle, or the TCA cycle, tricarboxylic acid cycle, because it involves tricarboxylic acids, or the Krebs cycle, because Hans Krebs elucidated this cycle based on pigeon muscle tissues. You generate reducing power through 2 oxidative decarboxylations, and then additional reducing power through a couple of other reactions. You generate free energy in the form of GTP, which is equivalent to ATP. You go from a 2-carbon and a 4-carbon molecule to a 6-carbon molecule, subsequently to a 5-carbon molecule, to a 4-carbon molecule, and you regenerate the starting material so that you can go around the cycle again. So the citric acid cycle starts with acetyl coenzyme A, which feeds into the cycle. It condenses with oxaloacetate to make a 6-carbon molecule of citrate. Okay, so 2 carbons enter as acetyl coenzyme A. 2 carbons leave as carbon dioxide through oxidative decarboxylations. The important thing to remember is that the 2 carbons that leave are not the same 2 carbons that came in as this acetyl group. They are 2 carbons from the oxaloacetate molecule. During the course of the reaction, you generate 1, 2, 3 3 NADH molecules, 3 reduced molecules of NADH, and each of those can subsequently be used to generate approximately 2.5 ATPs through oxidative phosphorylation, and we're going to talk about oxidative phosphorylation next. There's a single substrate level phosphorylation during this reaction in which you make a GTP molecule, and a GTP is essentially the same as an ATP from an energetic perspective. You also make an FADH, which makes 1.5 ATPs when it donates its electrons to the electron transport chain for ATP production through oxidative phosphorylation, and we're going to discuss why NADH is more energetically useful than FADH in the electron transport chain. And so it's a cycle. So at the end of the cycle, you regenerate the same 4-carbon molecule that you started with, and it's available as a substrate to pick up the 2 carbons of acetyl coenzyme A to repeat the cycle. Okay, so let's break this down, and we'll look at each of the individual reactions on their own. So on the final exam, I will give you a stripped down version of this, which just includes the structures of all the intermediates in the TCA cycle. I won't give you the names or any of the necessary cofactors, but I'll provide you with the structure of citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. The first reaction in the pathway is an isomerization, where you move the hydroxyl group from carbon 3 to carbon 2. Then you do the oxidative decarboxylation, as I explained to you. You lose a CO2 molecule and use reducing power to generate a reduced NADH. The next reaction is a second oxidative decarboxylation, where again you lose a CO2 and generate reducing power in the form of NADH. So you've gone from 6 carbons in citrate and isocitrate to 5 carbons in alpha-ketoglutarate to 4 carbons in succinyl-CoA. Remember that this thioester bond is a high energy bond, so upon its hydrolysis you can use the free energy that's released to convert a nucleotide diphosphate to a nucleotide triphosphate, and the coenzyme A is lost. We then generate a symmetrical molecule, succinate, where carbon 1 is exactly the same as carbon 4 and carbon 2 is the same as carbon 3. This is a substrate for succinate dehydrogenase, and the reducing power of the oxidation of this molecule is used to reduce FAD to FADH2. The FADH2 can then feed into the electron transport chain. There's a hydration reaction where a fumarase adds a water molecule across this double bond to generate malate. Malate you've seen before. It's a substrate for malate dehydrogenase, so this final oxidation event is linked to the reduction of NAD plus to NADH. And again, this NADH can be used in the electron transport chain. And we've regenerated oxaloacetate, which is a substrate for the aldol condensation catalyzed by citrate synthase to make a new molecule of citrate to continue through the TCA cycle again. So let's look at the reaction first, catalyzed by citrate synthase. Initially, these two active site residues are involved in general acid-base catalysis. This histidine residue acts as a general acid. This aspartic acid acts as a general base. This allows you to initiate the reaction. So first of all, the two substrates have to bind at the appropriate orientation in the active site for these residues to play their roles in general acid-base catalysis. Once the reaction is initiated, you get this enol formed, and this enol allows you to generate a new carbon-carbon bond to take this 2-carbon molecule of acetyl-CoA and condense it with this 4-carbon molecule of oxaloacetate. And again, in this case, the histidine is acting like a general base and pulling that proton off, and the aspartic acid is a general acid and donating a proton. So all we have to do now is to remove the CoA group, so cleave this thioester bond. This I'll show you in the next slide, but we can only do this after the citral-CoA is formed. What you don't want to do is cleave this bond in acetyl coenzyme A, because then the free energy would be lost and not captured. What we want to do is generate this 6-carbon molecule before we cleave this bond so that we can capture that free energy. So this is citrate synthase. So a synthase catalyzes a condensation reaction, in this case fusing a 2-carbon molecule with a 4-carbon molecule, but you do not require any ATP or any other nucleotide triphosphate to do this reaction. Okay. This reaction goes through this citral-CoA complex. So this citral-CoA is a transient intermediate. So oxaloacetate condenses with acetyl-CoA to make this transient enzyme-bound intermediate, and this enzyme-bound intermediate is then hydrolyzed with a water molecule to release coenzyme A and generate the product citrate. Okay, it's this hydrolysis step that keeps this reaction energetically favorable. So the CoA ester is a high-energy intermediate. Okay, the enzyme in us, okay, is a dimer, okay, and it undergoes induced fit. And the induced fit, or the conformational change upon oxaloacetate binding, excludes water and arranges the active site residues such that acetyl-CoA can bind, so that the oxaloacetate can interact with the acetyl-CoA to generate the citral-CoA before hydrolysis occurs. Okay, so the mammalian citrate synthase. So our citrate synthase is a dimer, and it undergoes a major conformational change during the course of catalysis. So induced fit. Upon OAA binding, you get a structural rearrangement that leads to the formation of the acetyl-CoA binding site, so that the enzyme can bring the two substrates close together in the proper orientation and allow certain bonds to be polarized, so it can carry out the chemistry. Okay, so induced fit upon the binding of the first substrate. So this allows the enzyme to hydrolyze citral-CoA but not acetyl-CoA, because if you simply hydrolyze the acetyl-CoA, that would be a waste. You'd lose the energy from the thioester bond. So what you don't want is the acetyl-CoA to bind until you've already got the OAA, the oxaloacetate, bound and in the catalytically appropriate orientation. Okay? So the induced fit prevents the undesirable side reaction. So similarly to how hexokinase prevents wasteful hydrolysis of ATP, citrate synthase doesn't allow you to cleave the thioester bond until you're ready to do it to get the best result out of the products. So the next enzyme is aconitase, which converts citrate to isocitrate. So aconitase simply catalyzes a dehydration and a subsequent rehydration. You could imagine that the same water molecule that comes off is the same water molecule that goes back on, but it doesn't have to be. So we're doing an isomerization reaction to convert citrate to isocitrate. And so why are we doing this reaction? That will become obvious in the next steps, okay? Effectively, we're converting this into a molecule that would be able to undergo the subsequent oxidative decarboxylation. Okay? So this enzyme contains an iron-sulfur center that allows you to run this reaction forward. The iron-sulfur center plays a role both in substrate binding and in catalysis. Adding the water molecule to cis-aconitate could give you either citrate, so you could simply go back in this direction, or isocitrate going in the forward direction. Citrate, under standard conditions, citrate formation is favored, but in the cell, you generate isocitrate, and by now, you should appreciate how that happens. So, iron sulfur center, so citrate synthase, so aconitase, contains the 4 iron 4 sulfur center. So it is a non-heme iron protein. So it's a protein that has required irons, but these irons are not associated with a heme. So acetyl-CoA attaches to oxaloacetate in citrate synthase to generate citrate. This is then a substrate for aconitase. It goes through this cis-aconitate intermediate and subsequently to isocitrate. And while under standard conditions the reaction would favor citrate formation in the cell, citrate actually gets converted to isocitrate, and by now you just should appreciate why this is the case. Okay? So if you look at citrate, it looks like a symmetrical molecule. But from the perspective of the enzyme, these two groups are not equal. Okay? So while this is not a chiral carbon, it has prochiral character. And that's because of the arrangement of space of these groups with respect to where they bind on the active site of the enzyme. Okay? So these groups must interact in a specific way at the active site. And there's no way you can rotate this molecule so that this group can replace this group in these weak interactions with the active site of the enzyme. So this positions the molecule so that only one group is susceptible to the reaction and not the apparently identical other group. Okay? So although this is not a chiral center, this molecule has prochiral character because of the way these atoms are arranged in space and how they must interact with the active site residues to carry out the reaction. Sorry, this one. The active site residues to carry out the reaction. Okay, so we've now generated a molecule of isocitrate. Now isocitrate dehydrogenase catalyzes the first oxidative decarboxylation reaction, which generates NADH and the 5-carbon molecule of alpha-ketoglutarate. So isocitrate, NAD plus gets reduced to NADH. This molecule gets oxidized from a COH group to a CO group. This makes this CO2 group a good leaving group. It comes off, and we've converted isocitrate to alpha-ketoglutarate. So we've gone from 6 carbons to 5 carbons by releasing one molecule of CO2. This carbon dioxide started as part of oxaloacetate, not as part of acetyl coenzyme A. Okay, so the 2 carbons that come in as the acetyl-CoA are not the 2 carbons that are lost in the first 2 oxidative decarboxylations. Okay, so this is why you need aconitase. You need aconitase to put the OH group here as opposed to here, so that you can carry out this conversion of a hydroxyl to a carbonyl to allow this carbon dioxide to be a good leaving group. Okay, so this reaction, okay, so isocitrate dehydrogenase. Anything I need to say about this? Oh yeah. So you've already seen a reaction that requires a steady supply of NAD plus to take place. Okay, in glycolysis, the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase needs a steady supply of oxidized NAD plus to work. Okay, if you don't have available NAD plus, then like the glyceraldehyde 3-phosphate dehydrogenase reaction, this isocitrate dehydrogenase reaction can't work. Okay, so this enzyme is actually the rate limiting step in the TCA cycle. So we've done our first oxidative decarboxylation. Now we'll do a second oxidative decarboxylation. So we're converting alpha-ketoglutarate into succinyl-CoA. Again, CO2 group is lost. We move from a 5-carbon molecule to a 4-carbon molecule. So it's catalyzed by the alpha-ketoglutarate dehydrogenase complex. Again, we're generating a molecule of NADH, and a molecule of CO2 is lost. This reaction is virtually identical to the reaction catalyzed by the pyruvate dehydrogenase